everybody. Today we are going to talk about a special type of osmosis, or a special type of diffusion, called osmosis. And so if you look in your notes, it says a special case of water, movement of water across the cell membrane. That is what we are talking about today. All right, so osmosis is really just the diffusion of water. Water is very important to life, so we have a separate way of talking about water. I'll move this out of the way so we can see the picture. So water diffuses just like anything else. Water diffuses from high to low concentration, just like any diffusion. It's going to go from high to low, except this is a high concentration of water to a low concentration of water. And this would be across a semi-permeable membrane. So we would be talking about water moving across a membrane that allows water to go through. Does anybody remember the name of the channels that allows water to flow through? Those channels were called aquaporins, and that's because water can't really very easily get through the bilayer without them. So the direction of osmosis is determined by comparing the total solute concentration. Now, you're going to have to keep very careful track of whether we're talking about solute or whether we're talking about water. So make sure you understand the difference between those as we go through this. So we are going to be comparing solute concentrations to help tell us how osmosis is going to work. There are three terms which describe a solution. Hypertonic, so the hyper means above, and that is more solute. So that is a solution with more solute and less water. Hypotonic, hypo means below, less solute, more water. And then we have isotonic means equal solute, equal water. So if we look at the picture here at the bottom of the screen, we can see that hypertonic is on the right and let me write. We can see that hyper is on the right, and that's because there's clearly more solute there. Hypo is on the left, that's because there's less solute. So now let's think about this in terms of water. Which side has the most water? The most available water is going to be on the left side, right? Because there's less water surrounding the solute. So which way is the water going to go? The water is going to go from a high water concentration, which is over here, right? There's lots of water, lots of available water on the left. And then there is a low water concentration on the hypertonic side. So water goes from high to low. Cells depend on the intake or loss of water for survival. So different things can happen. If too much water enters an animal cell, we can see in this first picture that the animal cell is going to lyse or break open and die. Isotonic is normal for an animal cell, and a hypertonic solution might cause an animal cell to shrivel. Meanwhile, for a plant cell, normal is when there is lots and lots of water coming in. And in an isotonic solution, that's sort of not ideal, not great, but really bad is a hypertonic solution where the cell is plasmolyzed because it has lost so much water. And we'll talk about what causes that to happen in a little bit. So a hypotonic solution would be fresh water. An isotonic solution is balanced. And a hypertonic solution is going to be something very salty or very sugary. All right, so let's first talk about hypotonic. Hypotonic would be a cell in fresh water. And that would mean that there's a high concentration of water around the cell. So what does that really mean? So if we're talking about a cell that's hypotonic, then that means if this is my cell and there's stuff dissolved in the cell, and surrounding the cell, if the solution is hypotonic, the solution has less stuff dissolved, right? So water is going to go from a high water concentration to a low water concentration. And And that means that it's going to go into the cell. A good shortcut for thinking about this is that the water is going to go to the area with a higher solute concentration. So water goes from high to low, and that means it goes into the cell. So if this cell was in fresh water has a very little stuff solute dissolved in it, so water is going to go in. Now, think about what happens when water goes into a cell like a water balloon. What's going to happen? It's going to swell up and get bigger. And a big problem is the cell can swell and burst. So we don't want animal cells in hypotonic solutions. A good example of this would be the paramecium. A paramecium is a eukaryotic cell that exists in fresh water. And what happens is that because we know that this cell can burst, we've got a problem, right? It's in fresh water, so water is continually entering the paramecium. And so what's going to happen? Is the paramecium just going to die and burst? No, the paramecium has a contractile vacuole, and the contractile vacuole is a specialized tool that basically continually pumps out water. That takes a lot of energy, so a lot of energy is going to be used by that contractile vacuole to constantly pump out that water. So there's our energy always being used. The paramecium has to spend a lot of energy maintaining homeostasis, maintaining this balance inside of it. Plant cells also have a handy way of dealing with hypotonic solutions, that the plant cells, when they become full of water, they've got their cell wall, and that cell wall is going to protect them from bursting. So that's a good thing for plant cells. So here we notice that a plant cell is turgid. Even though water is entering in this hypotonic solution, it's turgid and not lysed. All right, moving on. The plant cell is very happy, the animal cell is not. Here's an example of a contractile vacuole here, taking in water and then squeezing it out, helping maintain homeostasis, using lots of energy to maintain that homeostasis. Okay, hypertonic, if you remember, hyper means below. So, hyper means above, not below. Hyper means above. So a hypertonic solution would be a cell in a very, very salty or a very, very sugary solution. So cell in salt water, where there is a low concentration of water around the cell. So what would this look like? Here's my cell. My cell has a little bit of solute inside of it. However, around it, tons and tons and tons of solute, lots of solute surrounding the cell. So remember, we want to go from a high water concentration to a low water concentration, or another way of saying that is the water is going to go to the saltier area. The saltier area is on the outside, so water is going to travel out of the cell. All right. So here's the problem. The cell loses a lot of water, and then it can die if it doesn't have enough. An example of this is shellfish. Shellfish, if they are in very salty water, they can die. And what they do is they have pumps that can take in water that is being lost, or they can pump out salt. So they can maintain homeostasis by pumping different things in or out of their cells. Plant cells, if they are in a hypertonic solution, then that means they are going to undergo plasmolysis, a term you should know. That just means to wilt. We have a picture of that here. The cell membrane has pulled away from the cell wall. This would be a wilted plant, and it can recover. Same thing here with an animal cell losing water, and hopefully it can recover as well. All right. hyper, great. And then there's an isotonic solution, and an isotonic solution means equal. So this is when cells are immersed in a mild salt solution. So here we have a cell, little bit of salt in them, and about the same amount outside. No difference in the concentration of water between the cell and the environment. All right. So the problem here is none, especially if you're an animal cell. This is the type of solution you prefer to be in, right? What this means is that water is going to flow into the cell because water is always moving, but water is also flowing out of the cell at about the same rate. So we never really gain or lose any water if our animal cells are in isotonic solutions. This is how animal cells prefer to be. In a plant, it's the same thing, not preferred. Plants would prefer hypotonic environments, but they are fine usually in isotonic environments. There's no net movement of water. Keep in mind, there's always going to be movement of water. This is just no net movement. So in a hypotonic, the net movement was in. In a hypertonic, the net movement was out, but there was still movement in both directions. The flow of water is equal across membranes, and the cell is in equilibrium. And therefore, the volume of the cell is stable. It's not shrinking, it's not growing, it's not going to lyse, it's not going to become turgid. So our blood cells are in blood plasma, and that is an isotonic solution. And when you get an IV, it's also going to make an isotonic solution so your blood cells don't change too much. All right, so that cell is perfect, and the plant cell could be better. And this is all dependent on aquaporins. Aquaporins, they're numerous, and they can allow for rapid movement of water in and out of cells. When we do our osmosis lab, and when we look at it, you will see that it can happen very quickly. So in the early 90s is when aquaporins were discovered. Right? So the cell has 0.05 molar. The environment has 0.03. So the cell is hypertonic because it has more dissolved substances. Beaker is hypotonic because it has fewer dissolved substances. So if we are in a hypotonic environment, water is going to go in to the cell. Notice when we say the solution is hypotonic, we mean the solution of the environment is going in, and the cell could burst or swell. All right, and that is all for now. Please let me know if you have any questions about osmosis.